Hi, I'm joined today with Dr. Raphael Gübeli, who is Head of Marketing at c-LEcta, to talk about the current trends within the cell and gene manufacturing space. So welcome, Raphael. Delighted to have you here today. Could you please just begin by talking about what c-LEcta are doing in the space at the moment and your involvement within the biopharma industry?
Yeah, sure. Thank you. c-LEcta, we are a German company. We are founded in 2004. So next year we are approaching 20 years, and we are a manufacturer of GMP enzymes. So we are developing and manufacturing GMP enzymes for biopharmaceutical manufacturing. Since 2022, we have a majority shareholder, the Carry Group, which gives us long-term stability and also helps us to reach the industry around the globe. At c-LEcta itself, we are around 130 people, so a decent size. And in the cell and gene therapy space, what we are mostly known for is the GMP endonuclease called DENARASE. So DENARASE is used in viral vector manufacturing to degrade unwanted host cell and plasmid DNA. Pretty standardized process. And what does really differentiate us? That's a good question. We focus on highest quality and cost-efficient manufacturing and cost-efficient pricing for our customers, because we believe by doing that we can help the industry to grow and reach as many patients as possible. In the meantime, when we look at DENARASE, we are really excited because we have customers around the globe applying our enzyme in their preclinical and clinical up to commercial manufacturing. So we have hundreds of projects. We have already 4 medicines who are approved. In the manufacturing of these medicines, DENARASE is involved. So exciting times ahead.
Yes, definitely. And in terms of the challenges within manufacturing viral vectors for cell and gene therapies, what are you doing to sort of address these challenges?
As a scientist, the first thing that comes to my mind is always the technical challenges. And there's an industry discussion about technical challenges related to, for example, the yield of viral vectors. Common numbers that we hear is that the yield after purification is maybe 10 or 20%. So there's a lot of room to grow there. Some challenges to solve. Secondly, very important for viral vectors is the full empty capsid ratio. So also that a lot of companies, a lot of developers are trying to increase that ratio. Our GMP enzymes do indirectly help our customers to reach these, to solve these technical challenges. So we can have detailed discussions about, we can give them some consultancy when it comes to applying the GMP endonuclease in their manufacturing space and optimize it. Besides technical challenges, where we are oftentimes involved is when it comes to challenges related to supply. So we all remember the COVID times, huge pressure on the supply chain. Also, the supply chain of endonucleases was under pressure. We are proud to say from our side, we always managed to deliver on short to mid-term significant quantities. Now, in the meantime, the situation has clarified. The supply challenges are not as difficult to handle as compared to during the pandemic. However, what we did is we tried to learn from our hard work during the pandemic, and we tried to make our whole manufacturing and supply organization more resilient. We built up additional safety stocks because we believe that in the next 3 to 5 years, we will see a lot of viral vector-based gene therapies and gene-modified cell therapies getting approved and reaching patients. And that certainly needs much more manufacturing capacity and in essence also additional endonuclease volumes. So we are basically preparing for that, learning from COVID times.
Yes, thank you.
And maybe a last topic that is often discussed in the viral vector manufacturing space, a challenge, is certainly the cost. At the moment, viral vector-based therapies are reaching only a few patients, and I think everyone would like to extend that so that it can benefit additional patients. The challenge is that the manufacturing is very costly, and with our cost-efficient enzyme solutions and our support that we give to the developers, we can really make a difference. There are sometimes numbers in viral manufacturing. If CDMOs look at their costs of manufacturing viral vectors, they sometimes end up with 10 to 20% of the raw material costs being attributed to endonuclease. So you want to carefully look at that cost, and if we can help to make a difference, we're happy to do so.
Okay, great, thank you. And then in terms of sort of quality and regulation, how important and how does this affect the sort of production and manufacturing of cell and gene therapies?
Like all pharmaceuticals, quality and regulatory aspects are key. At the end, it's about safety for patients. When it comes to raw materials for viral vector manufacturing, but also in general about raw materials for biopharmaceutical manufacturing, it's, I think, in the meantime, common industry knowledge that one needs to source these raw materials in GMP grade from the suppliers who can offer it in a GMP grade early on in development, early on in preclinical trial manufacturing, in order to be sure that you select and you develop your process with the right materials that can then later on be used in GMP manufacturing for clinical trial and hopefully also for approved medicines. Starting early with discussions or with GMP grade material can really help you to solve the challenges or to solve the regulatory questions. From a supplier perspective, we see certainly a strong increase in regulatory and quality questions from our customers around the world. And we're happy to solve that together. Because at the end, it helps our customers, but it also helps us to anticipate the future needs in terms of quality and regulatory and to further improve our portfolio and our service. And yeah, from a quality and regulatory aspect, it's all about transparency. It's about making sure that you can discussing with the developers about and the quality people about release procedures of your raw materials. It's important that you can be transparent, and that's what we try to do with our products and our interactions.
And in terms of CDMOs, how important are these and why in the development of cell and gene therapies?
There are numbers in the industry saying that 60 to 70% of the manufacturing capacity is at CDMOs. So I think it's fair to say that CDMOs do have a systemic relevance. In the last couple of years, there was always not enough capacity at CDMOs. So there were long queues of projects that needed to be manufactured at CDMOs. In the meantime, that has normalized a little bit due to the fact that a lot of CDMOs also expanded their capacities in viral vector manufacturing. And that's really a good time now, because the more therapies we see going through clinical trials and the more therapies get approved and reach patients, the more manufacturing capacity we need. And at that time, it's very crucial to have enough. So it's interesting to see that. We also see that CDMOs offer or develop their own platform processes in order to help their customers to embark on a CDMO with their specific project with their specific process. And we are working as a supplier of GMP enzymes with quite some of these CDMOs to make sure that they know they can define the design space with the GMP endonucleases, for example, and that they also get the forecast and supply that they need.
Mm-hmm.
Yeah.
Mm-hmm. And just finally, where do you see the next 5 years taking the cell and gene therapy space, and what are c-LEcta doing to support this evolution?
The next 5 years will be super exciting. On the one hand side, we will see a lot of gene and cell therapies making it to the approval, hopefully. So we will see many more patients benefiting from it. We will see the industry growing with that. And then what really excites us too is that apart from viral vectors, there's a lot of innovation about additional delivery methods for genetic medicines, being it mRNA in lipid nanoparticles, being it CRISPR-Cas, ribonucleotide complexes. That's super exciting because we believe that these technologies, these delivery technologies, can further broaden the application of genetic medicines and treatments for additional diseases and additional patients. And from a perspective as an enzyme manufacturer, it's also exciting because most of these new delivery methods, the manufacturing of them also needs, doing the manufacturing, you also need enzymes. So we are super excited. We have quite some products in our development, and you can be sure that we will be launching some interesting GMP enzymes fitting the new innovative delivery methods. So
very exciting.
DENARASE is just the start of a long journey for our company.
Oh, great. Well, thank you so much for joining us today. It's been a pleasure speaking with you.
Thank you.